Dear students, Assalamu alaikum. I'm Kiran Fatma and I would like to welcome you back to our series of programs on biology. The topic of discussion is physiology with a particular emphasis on the conduction of the nerve impulse and the physiology of the muscle tissue. Since we are going to discuss the physiology of the muscle tissue, it's important to recall our concept of coordination. This information will help us better understand the complex mechanism of conduction of nerve impulse. You may be aware, having read about the different types of living organisms found on Earth, that as you climb up the ladder of evolution, there is an increased trend of division of labor among cells and tissues that forms organs, that form specialized structures, whose function is integrated with each other and regulated by various mechanisms. You know that integration means unifying different activities to different things in order to reach a specified goal. In our case, it may be digestion of food, response to a stimulus, or nervous coordination, that is conducting a nerve impulse. Organization means working together in a regular manner so that timing and relationship result in a smooth and effective activity. All these activities can be achieved by a process which is known as coordination. Coordination is the remarkable ability to maintain a stability in the internal environment, which is called homeostasis. In short, we can say that coordination is brought about by the nervous and endocrine systems. Let's first discuss about the nervous coordination. The functional unit of nervous system are known as neurons or nerve cells. Neurons are specialized to produce signals that can be communicated rapidly and precisely to other cells. The nervous system of snail, frog, human, or any other complex animal is constructed through three types of neurons: sensory neurons, intermediary neurons, and the last one is motor neurons. Sensory neurons serve as the receptors or are activated by the receptor tissue organ. They are stimulated by changes in the external or internal environment and respond by relaying signals to integration centers such as the spinal cord. Intermediary neurons or interneurons are located in these integration centers, which interpret the information arriving on the sensory lines and then influence other neurons in response. The third type of neuron is the motor neuron, which relays information away from the integrating centers, and these integrating centers are the brain and spinal cord. The motor neurons relay information away from these integrating centers to the body's effector tissue organs such as the muscles and gland cells. Neurons come in various sizes and shapes, but they all have three parts: dendrites, axon, and cell body. The dendrites, which has come from the Greek word dendro, means tree, receives information from other neurons and generally conduct nerve impulse towards the cell body. The axon, which means axis, conducts nerve inputs away from the cell body. The cell body contains the nucleus and other organelles typically found in cells. One of the main functions of the cell body is to manufacture neurotransmitters, which are chemicals stored in the secretory vesicles at the end of axons. The important point to note is that the dendrites and axons are sometimes very long. They are then referred to as fibers. The dendrites and axons in higher animals are covered with white myelin sheath, as you can see in this diagram. The neurons are grouped together to form a nerve. Now we come to the electrochemical phenomenon, involving the movement of unequally distributed ions on either side of a neuron plasma membrane. Later, Hodgkin and Huxley, the English neurophysiologists, proved this to be true when they managed to insert a tiny electrode into the giant axon of the giant squid, as you can see here in this diagram. The squid axon produces rapid muscle contraction so that the squid can move quickly. The squid was selected for the experiment because its axon is so large, it is about 1 millimeter in diameter. So it is convenient to put a microelectrode in it. When the axon is not conducting a nerve impulse, the electrode registers and the oscilloscope records a resting potential of 65 MV, as you can see in this figure. Just prior to an axon conducting a nerve impulse, the threshold for an action potential has been achieved, followed by a rapid change in potential from 65 MV to 40 MV, which is known as depolarization, followed by a return to 65 MV, which is known as repolarization. You can see here in this figure the rise of potential from 65 MV to 40 MV. The details of the action potential and resting can be well explained with the figures which are given here. The action potential is the result of an exchange of sodium ions and potassium, and it is shown as a change in polarity by an oscilloscope. When a neuron is not conducting a nerve impulse, the sodium and potassium gates are closed, as you can see here in this figure. The sodium-potassium pumps maintains the uneven distribution of these ions across the membranes. You can see in the figure very clearly the oscilloscope registers a resting potential of 65 MV inside compared to outside. An action potential begins when the sodium gates open and sodium ions move to the inside. It is very clear in this diagram the movement of sodium ions from outside to inside. The oscilloscope registers a depolarization as the cytoplasm reaches 40 MV compared to tissue fluid. You can see in this graph registering depolarization by oscilloscope to 40 MV from 65 MV. The action potential continues as the sodium gates close and the potassium gates open, allowing potassium ions to move to the outside. The oscilloscope registers repolarization as the cytoplasm becomes 65 MV as compared to figure fluid. The oscilloscope registers 65 MV again as the sodium-potassium pump works to restore the sodium-potassium ion distribution. The gates of sodium and potassium are now closed but will open again in response to another stimulus. When the axon is not conducting an impulse, the oscilloscope records a membrane potential equal to about 65 MV, indicating that the inside of the neuron is more negative than the outside. Once again, you see this diagram which shows the above statement. This is called the resting potential because the axon is not conducting any impulse. Dear students, now we come to the second part of our program, that is the physiology of the muscles. We'll take the example of the skeletal muscles and study their structure and action. Each skeletal muscle is composed of many muscle fibers that contract when they are stimulated by the motor neurons. Each motor neuron branches to integrate a number of muscle fibers, and all of these fibers contract when their motor neuron is activated, to give rise to a coordinated contraction by all muscle fibers. The number of neurons activated determines the gradation in the strength in the contraction of the whole muscle. For example, for a heavy loader activity, many neurons are activated, whereas for a light loader activity, only few neurons are activated. Through these diagrams, you can see the action of skeletal muscle. In this diagram, you can see the extensor that increases the angle at a joint whereby the knee straightens. The flexor, on the other hand, decreases the angle at a joint as shown in this figure. The abductor moves the limb towards the middle of the body. The abductor moves limb away from the midline of the body. Other categories of muscle actions include the elevator moves insertion upward, whereas depressor moves insertion downward. The rotator rotates the bone and the sphincter constricts an opening. As a result, the opening either narrows or is completely closed. In all cases, the skeletal movements are produced by the shortening of the appropriate muscle groups, the agonizers, while the antagonist remains relaxed. The fibrous connective tissue proteins within the tendon continue an irregular arrangement around the muscle to form a sheath which is known as epimysium. Connective tissue from the outer sheath extends into the body of the muscle, subdividing into columns or fascicles. Each of these fascicles is thus surrounded by its own connective tissue sheath known as perimysium. A muscle fascicle is composed of many muscle fibers or myofibers. Each is surrounded by a cell membrane or sarcolemma, enveloped by this connective tissue layer called endomysium, the muscle cells. Since the connective tissue of the tendons, epimysium, perimysium, and endomysium is continuous, muscle fibers do not normally pull out of the tendons when they contract. Despite their unusual fiber shape, muscle cells have all the organelles that are present in other cells, such as mitochondria, glycogen granules, and intracellular membrane, and others. But unlike most other cells, muscle fibers are multinucleate. The most distinctive feature of the microscopic appearance of skeletal muscle fibers, however, is their striated appearance. The striations are produced by alternating dark and light bands that appear to cross the width of the fiber. The dark bands are called A bands and the light bands are called I bands. At high magnification in an electron microscope, thin dark lines can be seen in the middle of the I bands. These are called Z lines. The labels A, I, and Z are useful for describing the functional architecture of muscle fiber and were derived during the history of muscle research. The letters A and I stand for anisotropic and isotropic respectively, which indicate the behavior of light as it passes through the regions, whereas the letter Z comes from the German word Zwischenschieb, which means between discs. In this diagram, it is very distinctly explained a muscle fiber contains many myofibrils, sarcolemma, a band, I band, and Z line. While discussing the mechanism of contraction, you must keep in mind that the A bands within each muscle fiber are composed of thick filaments and the I bands contain thin filaments. Movement of cross bridges that extend from the thick to thin filament causes sliding of the filament. Here in this diagram, you can see that over the thick filament, the thin filament is sliding. As a result of this sliding of the filaments, the muscle tension and shortening results. The activity of the cross bridge is regulated by the availability of calcium, which is increased by electrical stimulation of the muscle fiber. Electrical stimulation produces contraction of the muscle through the binding of calcium to regulate proteins within the thin filaments. When muscle cells are viewed in the electron microscope, each cell is seen to be composed of many subunits known as myofibrils. These myofibrils are approximately 1 micrometer in diameter and extended in parallel rows from one end of the muscle fiber to the other. The myofibrils are so densely packed that other organelles such as mitochondria and intracellular membranes are restricted to the narrow cytoplasmic spaces that remain between adjacent myofibrils. In this diagram, you can see the positions of the mitochondria and nucleus, which is in the periphery of the fiber near the sarcolemma. When a myofibril is observed at high magnification in longitudinal section, the A bands are seen to contain thick filament. It is these thick filaments that give A band its dark appearance. The higher I band, by contrast, contains thin filament. The thick filaments are composed of the protein myosin and the thin filaments are composed primarily of the protein actin. We continue our discussion on the mechanism of muscle contraction. The most convincing and important theory of muscle contraction is the sliding filament theory of contraction. According to this theory, when a muscle contracts, it decreases in length as a result of the shortening of its individual fibers. Shortening of the muscle fibers, in turn, is produced by shortening of their myofibrils, which as a result of the shortening of the distance from Z line to Z line. As the sarcomere gets shortened, however, the A bands do not get shortened but instead move close together. The I band, which represent the distance between A bands of successive sarcomere, gets decreased. In this diagram, you can see that the sarcomere is relaxed. The distance between the I bands should be a point to be noted. In the diagram, you can see that the distance between the Z lines decreased and also the distance between I bands is also decreased. Sliding of the filaments is produced by numerous cross bridges that extend out from the myosin towards the actin. As you can see in this figure, the cross bridges are part of the myosin proteins that extend from the axis of the thick filaments to form arms that terminate in globular heads. You can see in this diagram the globular heads from the thick filament of myosin projecting towards actin. The orientation of cross bridges on one side of a sarcomere is opposite to that of the cross bridges on the other side, so that when the myosin cross bridges attach to the actin on each side of the sarcomere, they can pull the actin from each side towards the center. The action of cross bridge can be very effectively understood through this diagram of cross bridge cycle that causes sliding of the filament and muscle contraction. In this figure, the resting fiber is shown. Here you can see that the cross bridge is not attached to actin. The next diagram reveals that the cross bridge of myosin has just binded the actin. Following the previous diagram here, you can see that the par stroke causes filaments to slide. While here you can see that a new ATP binds to myosin head, allowing it to release from actin. While completing the cross bridge cycle, ATP is hydrolyzed, causing the cross bridge to return to its original orientation. Dear student, this phenomenon of sliding filament theory of contraction has more details involving some chemicals like calcium in muscle contraction. In fact, the attachment of calcium to troponin causes movement of the troponin tropomyosin complex, as it is shown in this figure, which exposes binding sites on the actin. The myosin cross bridges can then attach to actin and undergo a par stroke. With this, we come to the end of our program. Till we meet again, I wish you the very best. Allah Hafiz.